Okay, good afternoon, everyone. It's really a pleasure to be here in this session that is dedicated to the research for biotech and energy high-tech innovation. I would like to thank to Diego and to the organizer for this opportunity to chair this session. And it will be a really exciting session because we have many speakers and many in different fields. So I would like just to make a short introduction to this session and also present some of our new developments in the lab that I'm heading. But before that, I'm the head of Biotechnology Development Unit, if you don't know until now, and I'm working in the International Center for Genetic Engineering and Biotechnology here in Trieste, Italy. And it's my pleasure to present you Ronnie Andrews, that is my co-chair today. So he's a president and CEO of Oncocyte, from a company from California, USA. So please, Ronnie, if you want to say something.
I'll apologize in advance. My Italian is very poor, so I will be speaking English. Try to speak slowly, but very honored to be here. Thanks to Diego and the folks for inviting me. The presentations so far have been terrific. My company is synthesizing large-scale genomic data into smaller RNA panels that can affect the outcome of patients with cancer and solve the problem we have, which you'll see later in my talk, where we are spending somewhere around $120 billion in therapy in the US alone, probably about $300 billion globally, and less than 25% of that therapy has a durable response in cancer patients. And so we are spending a lot of money for the wrong reason. And so I left retirement to go back to work, to take some of the large-scale genomic data, synthesize it with AI and various mathematical instruments to create smaller panels that can actually help patients get the right drug at the beginning of the disease cycle. So it's an honor to be here, and I'm very much looking forward to our session. So,
thank you very much. Please, can you put the presentation on? Okay, so we will have 11 speakers, and I will try to be fast, and every speaker will have 10 minutes. So I would suggest to have a presentation of around 8 minutes, and then we can have a 2 minutes discussion and questions and answers. So something that I'm repeating myself from the May, because in May we had some satellite event on the HighBiotech, and we were talking about the Industry 4.0. So before I will present you the work of my group, I want just to remind all of us that the term industry, biotech 4.0, biopharma 4.0, refers to the fourth industrial revolution, which brings together rapidly evolving technologies such as Internet of Things, artificial intelligence, robotics, advanced computing to dramatically change the landscape of manufacturing, if we are talking about biomanufacturing. So it's characterized by integrated, autonomous, self-organizing production system. Why I'm saying that? Because I want to present you just a short overview of our activities in the lab when we are also trying to face this production process intensification when we are talking about biological drugs and biopharmaceutical medical products. We can utilize different modeling tools to better understand and guide different production process in a more efficient and knowledge-based way. So today, everything in the industry, biopharma 4.0 has to be better, smaller, easier, real-time, and more efficient, as we heard from Ronnie. So why biotechnology-derived drug products? Because FDA just approved 100 monoclonal, for example, monoclonal antibody product, because monoclonal antibodies are the biotechnology-derived drug products that are continuously gaining importance to the pharmaceutical industry. So it's very interesting to say that biological products are expected to account for a majority share in the top 100 drugs in terms of sale by 2026, with 55% of the total top 100 product sales in 2026, compared to 39 that was in 2012. The annual growth in global prescription sales of biological products will outpace conventional drugs between 2000 2019 and 2026. So in different therapeutic areas, so like Ronnie said, not just in cancers, but the treatment of different cancers were really revolutionized by entrance of monoclonal antibodies. Main producers are mammalian cells, Chinese hamster variant cells. Why I'm saying that? Because lately, last year, since we moved to the new labs, my group is working on the production of the trastuzumab biosimilar, that is recombinant humanized IgG1 monoclonal antibody that binds with a high affinity and specificity to extracellular domain of HER2. And so why? Because it's essential medicine for early stage breast cancer, metastatic cancer, and breast cancer is the most common cancer for women in both developing and developed countries, and the leading cause of cancer death for women. So a patent expired in EU 2014 and 2019 in USA. So now we are free to produce it as a generic of biological drugs, so-called biosimilars, and actually there are many big players, big pharma companies that now put the biosimilar trastuzumab on the market. We use it as our exercise. We wanted to see, can we do something about intensification of process and development of monoclonal antibodies in general, practicing on the first molecule like trastuzumab. It included a lot of expression trials, studies, CHO cell line engineering using CRISPR-Cas9 gene editing tool that we did in-house. We prepared the good CHO cells. We prepared the good expression vectors. And then when we come to this intensification and to be more efficient, we invested money in some automation. So the screening of the cell population for highest stable producing clones is a key rate limiting step in the development of these biotherapeutics. So my group adopted automation for high throughput clone selection using colony picker clone picks tool, so we can select the clones much faster with extracellular secreted protein in semi-solid media in a high throughput manner. So what does it mean? Just as I'm presenting here, so we can fastly from all these cells that are, white spots on this plate, you can easily pick up the green one that are producing, that are highly producers of the monoclonal antibodies. So we established this, and we have this all the line of the cell line generation scale, like starting that we succeeded to compact to the 4 weeks. So maybe before we needed for 1 year to do all this work. Today we can do it in as fast as 4 weeks. Then we did all the kind of the clone selection, amplification, and we arrived to the bioreactors, and we were able in 4 weeks to do it, to screen 17,000 clones to arrive to the high producer clones that we tested in the bioreactors. We developed the fed batch processes, and we arrived to the fast, very fast, from 2 milligram, as you can see on the top of this table, like from 2 milligram per liter production, we arrived just in the first round of this optimization and intensification process, we arrived to 1.2 gram per liter. So this is really good for starting point if you want to talk to the industrial partners. So I just came back from my tour from South America, where I visited 4 different countries, different companies and everything, and more like we are there. So we can offer the very efficient, very fast platform to produce and to develop the production of monoclonal antibodies or other recombinant proteins from the mammalian cell very fast. After that, we are also trying to adopt new technologies like the purification technologies. We are using the smart chromatography in trying to eliminate the clarification step in the production process, and we are now offering that to the companies as well, so that can save them time and money. And also during all this study, we also, of course, we checked quality control of the trastuzumab. We are able to perform all these quality control tests. We are a very small team, and we are very happy that in such a short time, as a non-profit international intergovernmental organization, we were able to do what big pharma is doing today. So we really showed that yes, we can work something on the intensification, and that having a more reliable and good process. So I hope I was very fast, but this is something just to update you with our thinking. And of course, all these things that we, as our session is a research for biotech, this is all research for biotech. And it's also research that we can now apply to any different field of research. So inside the Institute, we are trying to collaborate with different groups, cardiovascular group and neurobiology group, trying to see if we can use also this picker, colony picker for different purposes, and trying to intensify also their research for biotech. Thank you very much for your attention. So I would now go further with, I had a list, but I can just go this. So I would like to invite the first speaker, that is Alessandro Marcelo, who is the head of the Laboratory of Molecular Virology at the same center where I work, from the ICGB. Please.
English or in Italian, as you prefer. I know the chairman is not speaking Italian, so
I have converters, so if you want to speak in Italian, please do so.
Ok, allora proseguo in italiano, se vi va bene.
I'll keep on in Italian if you all agree. Good morning. My name is Alessandro Marcelo. I am a virologist and I work at ICGB here in Trieste. Many of you already know me. What I would like to talk about today is the type of research transfer we do here in Trieste and our member states. ICGB is an international center. It's no university or no academy or no industry. Its mandate is to transfer biotechnology in the interest of our countries. Our countries, as you see on the slide, are different countries in Africa, Asia, Latin America. Many of them are developing countries in need of low-cost and high-efficiency technologies. As Natasha before me already explained, our interest is to carry out research projects in this transactional framework. We work with diagnostic antivirals specifically, and we cooperate with the industry working in this field to find solutions for our countries. With the last 2 years and the pandemics, we've witnessed the technology and the research linked to virology enjoyed an important positive effect. In the last years, when I participated in these events on BioHighTech, I spoke about our activities within the frame of coronavirus and the antiviral diagnostical devices we developed. This year, I want to move a step forward or behind, depends on the point of view, and I'm going back to the virology in its complex, not only SARS-CoV-2, but all the viral options that we had before COVID-19 are still to exist afterwards, so we need to concentrate on this. But COVID-19 has accelerated many technologies, in particular, some diagnostic technologies we deal with, we currently operate in. The first one is a molecular technique that is similar to PCR, and it's called LAMP. It is an isothermal technique with no continuous temperature variation. It has a continuous temperature. It's easier to perform, but it is still very specific and sensitive as the PCR test. We've made clinical trials in 4 African countries with a technology New York Biolabs developed. They developed a kit for infrastructure, so we made some tests in Africa to prove that this technique was specific and as sensitive as the diagnostic standard, molecular standards, of course. The reading out of the technique was not optimized to facilitate the countries that need a simple samples that could be
Non
easily
collaboriamo anche
used
con
in their own
tecnologie più
laboratories.
sofisticate,
We
con
also work with
Halifax,
more
che è un'azienda
sophisticated
qui
technologies
del
with
territorio,
Halifax,
loro hanno
a
sviluppato
company working
un
here in the
sistema di
region. We've
PCR
developed
portatile
a
per la
portable
diagnostica molecolare
PCR
e noi abbiamo creato
test
dei
system
kit
for molecular
dedicati sia a
diagnostics and
SARS-CoV-2
created
e
chips
appunto, ma
dedicated
anche ad altri virus, in
to
particolare ci occupiamo
SARS-CoV-2,
dei
not
virus,
two, but
di
also to other
arbovirus, quindi
viruses,
virus portati da
in
insetti
particular
o zeppe,
arbovirus,
e abbiamo
airborne
sviluppato degli antitest
virus,
per
viruses
chikungunya,
carried by
settembre,
insects
sempre
and
applicabili a
other
questo sistema portatile di
species,
PCR.
which can be applied
Quindi
to
diciamo,
these
in
PCR
questi 2
portable
anni
system.
siamo anche
In the last 2 years,
attrezzati per svolgere trial clinici
we've been
focalizzati
equipping
a,
to perform clinical
testare la qualità
trials
di alcuni approcci
aimed
biotecnici
at
di diagnostica
testing
nei paesi,
and
nei nostri paesi, ma anche in
validating
Italia
some
abbiamo
diagnostic
testato sia
devices and
gli
systems
approcci di
in other
LAMP,
countries and in
sia
Italy.
approcci
For example, LAMP
diciamo di
approaches
test antigenici veloci
and
per un'azienda
rapid
cinese.
antigen
Quindi
tests with
la nostra attività
a
è quella
Chinese
di
company.
cercare
So our
di
activity
collaborare con laboratori o
concentrates
con
on
aziende
collaborating with
biotecnologiche per sviluppare
biotech
prodotti che abbiano
labs or
un'applicazione nei
companies
nostri
to develop
paesi
products that can be applied in
membri.
our countries, in our member
Questo
countries.
lavoro ci ha portato a collaborare con la Fondazione Gates
This
per
kind
un
of work
progetto, diciamo, in
led
cui
us to
questo
cooperate
approccio
with
di
the
diagnostica
Gates
per
Foundation
SARS-CoV-2 viene
with this
ampliato a 10
diagnostic approach
paesi
for
in Africa
SARS-CoV-2
e lo stiamo
has been extended to
sviluppando i
10 countries
protocolli
in
per
Africa. We've
portarlo dai
been
tamponi,
developing
diciamo che
protocols to
classici della diagnostica
transfer
a
it
test
from
salivari diretti,
classical
quindi
swabs
più
to
semplici e
saliva, direct saliva
meno
tests
costosi dei test diagnostici
that are
classici.
simpler
E quindi
and
ci stiamo
less
attrezzando
costly
per
than
poter
diagnostic,
portare questa
classical
tecnologia
diagnostic
in
tests.
10
So we are
paesi
trying to equip
africani.
ourselves to bring this technology
Come
into
dicevo, non ci
10
occupiamo solo di
African
diagnostica,
countries.
ci
As I
occupiamo
was
anche di
saying,
antivirali
we
e
do not only deal
stesso
with
discorso per la
diagnostics, but
diagnostica,
also
con il
with
SARS
antivirals.
chiaramente anche c'è stato un forte impulso allo sviluppo di
With
antivirali, ma
SARS-CoV-2,
soprattutto, per quanto ci riguarda, di
we've
antivirali ad alto
experimented, as we
effetto.
said, a strong
Sapete
impulses
che
also for
gli antivirali in realtà disponibili sul mercato
broad
per
range
l'infezione virale non sono
antivirals.
poi tantissimi
You know
e
that
sono
we do
molto
not have
specifici
a
per alcune
large
infezioni.
quantity, a
Quello che
large
cerchiamo di fare
number
invece è
of
trovare
the antivirals that are
delle
disposal. They are
molecole, degli
specific
approcci che
for
siano più ad
specific
ampio spettro, che
infections.
quindi
We are
permettano
trying
di
to
controllare
find
l'infezione,
small
diciamo,
molecules
di
that can
classi di
control
virus che
infections
se si
of
presentassero in
different
futuro
classes of
avremmo a
viruses,
disposizione, diciamo, una batteria
which, if they were to
di
come back in the
antivirali
future,
già a
would enable
disposizione
us
per
to
contenere
have a battery of
un'eventualità futura.
antivirals suitable to contain
Qui c'era
in
un elenco
a
di
sanitary
molecole che
emergency,
abbiamo
a sanitary
testato negli
crisis.
ultimi 2
Here you
anni,
have a list of
li
the
abbiamo
molecules we've
illustrati in
tested in
collaborazione
the
con
last 2
BioValley
years.
per il trattamento del SARS-CoV-2, ma è un farmaco che può essere utilizzato anche
Miglostat in collaboration with
originariamente
BioValley,
era
a
stato
drug
scoperto come un inibitore di HIV.
that was
Non viene utilizzato come
originally
inibitore di HIV, è un farmaco che viene
discovered
utilizzato
as an
per le
HIV
malattie genetiche, quindi
inhibitor,
è un
and
caso
it
di
is
repurposing, quindi
now used
riposizionamento di
for
un farmaco,
genetic
ma è attivo anche
diseases.
con dengue,
It's a
contro dengue e contro
repositioning case
SARS.
of a drug. It can be used also
L'icosocosib
against
è un
the
suo
dengue
omologo
fever, for
anche attivo
example,
contro dengue e SARS.
chelgosivir.
Il 27 ossisterolo è un composto
27OHC,
sviluppato da
a
Panoxivir, che è
compound
uno spin-off
that was
dell'Università di
developed
Torino,
by
con cui
Panoxivir,
abbiamo
which is a
collaborato per
spin-off of the
dimostrare che è
University
attivo
of
contro
Turin, with which
SARS-CoV-2.
we collaborated.
Questo
We've
aveva già visto
collaborated
che
to
era attivo
um,
contro
develop a
endovirus, quindi
product
altri
against
virus sempre
SARS-CoV-2
respiratori, e
and
stiamo anche
other
cercando di svilupparlo
respiratory
e di
viruses,
portarlo avanti
and we
su tutti i
are
clinici.
trying to keep on
Su questo
developing
anche abbiamo
with
brevetti che sono
preclinical
pendenti
and
allo
clinical
stato,
testing.
di cui ho parlato
There's also a
precedentemente.
patent
La
pending on
nitroxalina
this.
è un antibiotico
Nitroxalin
che abbiamo dimostrato essere
is an
attivo contro SARS-CoV-2
antibiotic which is
e
active
qui
against
abbiamo in progress
SARS-CoV-2.
con un collaboratore in Slovenia un
Here
trial
we have a
clinico e
progress with a
anche in questo
clinical
caso
trial
abbiamo
in progress
un
with a
brevetto
Slovene collaborator, and
diciamo insieme
also
ai
in this
nostri
case, we have a
collaboratori a
patent
Lubiana.
together with our
In
collaborators
collaborazione con
in
Sun Pharma, che è
Ljubljana.
una biotech indiana,
Together with
abbiamo
Sun
contribuito
Pharma,
a
which is an
dimostrare
India
l'attività
pharmaceutical company,
antivirale
we've
anti-SARS-CoV-2, perché antidengue era già conosciuta, di un
proved
fitofarmaco,
the antiviral activity of
AQCH,
AQCH,
per il quale è stato fatto anche un trial clinico di fase 3 in
for
India
which
l'anno
we also
scorso,
held
i cui
a
risultati sono
Step 3 clinical trial in India last year. The results are good, but the trial is still ongoing, so the drug is not yet on the market. We also have other drugs, as you can see here, leverasidone, elbavicivir, which is an antiviral class, a broad-range antiviral that we have tested and for which we do not have intellectual property, but this can be pilot drugs to be used for further developments. So also in this case, collaboration projects with the industry or academies in our member state to bring about our antiviral research tests till preclinical trial. And of course, in some cases, we need to interface ourselves with the bigger, larger industries. To conclude, an aspect that I think is still undervalued, that is the containment of the infection of SARS-CoV-2 infection, is the activation of the virus in closed environment. We are all here, pretty calm till a couple of years ago. We couldn't even think about it, but basically the air circulation is an important vehicle for infection, not only for SARS-CoV-2, but also for other bacterial pathogens or viruses for respiratory diseases. This is true for conferences, this is true for the school, for cruises, cruise boats, many public contexts and environments. We need to have a protocol for treating the air and the surfaces that can still contain the infection. We launched a collaboration with Fincantieri a couple of years ago because they were strongly impacted by SARS-CoV-2 because of the cruise boats, because of the risks in closed environments. So we launched some air treatment systems for treating pathogens, but also using visible lamps produced by BioTech, which is an Italian company, and which can also be used in such rooms, such halls, where when you have many people all together, that act as antiviral devices, and we've proved their effectiveness in the lab. And these are other aspects of our collaboration with the industrial sector that needs to be better considered. To conclude, I would like to say that our projects are based on strong collaborations, as I said before, as the project with the Gates Foundation in Africa. We also have projects in Moldavia, a project that is funded by the region of Friuli Venezia Giulia to implement virology diagnostic protocols in Moldavia. Moldavia is becoming a very important country in this respect and in political terms, above all in this moment, in this very moment. We have a collaboration network with different European countries and Eastern European countries, Russia included, to identify and treat infections, tick-borne viruses and infection that is a problem affecting Eastern European countries, but also Italy and especially Northern Italy. This is an ongoing process, an ongoing project where we try and develop this kind of diagnostic approaches. Thank you for your attention.
Thank you very much, Alessandro. So we have just maybe time for one quick question or comment.
Just having spent my younger years in the world of HIV in Africa and working with PCR, I just want to reiterate one theme. I think the work you're doing to reduce the cost and increase the speed at which we diagnose, not just SARS-CoV-2. I think a silver lining of SARS, if there was one, was the acceleration of these rapid PCR technologies and the collaboration, and just congratulations. And it's something we in America need to do more of with our international partners.
Well, thank you. I appreciate the fact that you could follow my presentation for the translated. Yes, I mean, this is an effort that we are doing, we have been doing a lot in the past and has been accelerated a lot in the past 2 years. We are collaborating with New England Biolabs, which is an American company,
right.
with the Gates Foundation, which is American-oriented.
Right.
But they have really a will to favor diagnostics in Africa. And I mean, I think it's very forward-looking in a way that if you contain infections in those countries, you contain infection everywhere, and not only related to SARS, but for many other diseases.
Yeah, we learned that the hard way the last 2 years, so great work, and thank you.
Thank you.
Thank you very much. It's been very intensive 2 years for virologists, so congratulations. Our next speaker is Lavinia Alberi-Auber. She's director of the Swiss Integrative Center for Human Health from Freiburg. Lavinia, the floor is yours.
Thank you, and it's a pleasure to be here again in a more relaxed environment. And I will present again a little bit of what has been our journey since BioValley acquired us in 2001, and where we are in Switzerland, the landscape of Switzerland, but also how it is unique at the Institute. We have a strong focus on salivary diagnostic, what we have done in the past during the pandemic, and what has become our current activity in the service industry. And then I will talk a little bit about our research and development and also product developments. So we are a small for non-profit institution, private-public, with BioValley detaining 88% of the shares. And we are strongly focused on R&D for human diagnostics, trying really to democratize diagnostics and to find early therapies. So we are, as I mentioned, a public-private institution. So BioValley has 88% of the shares, and the remaining 12 is among those institutions, which are hospitals, universities, and also a center for biotechnology. We have 3 startups that are using our services and also using our service, and 2 are in the diagnostics and genomics and Alinova, and then we have also diagnostic, environmental diagnostic particle vision. So where we are in the western part of Switzerland. As you might have heard, I was speaking with my colleague in French, but actually we do speak 4 languages currently. And we have our partnering institutions are a little bit distributed across western Switzerland, also with the hospital in Ticino, and of course big in Trieste. So we really are strongly committed to bringing solutions which are accessible to human health, and of course we do have also a strong interest in discovering new solutions, and through R&D program that we can access through public funding. And then we also are selling the Chemo Maker in Switzerland and doing product development, both for the integration with the dedicato software from Becton & Dickinson and the integration into insulator, which are very common in the French-speaking part of the world. So as I mentioned, we are very, very focused on democratizing actionable diagnostic. We have reached about 30,000 customers in whole through Switzerland. Of course, Switzerland is a very small country, only 8 million inhabitants, but still we are a very, very small group, so 5 people reaching 30,000 subjects. It's, I think, pretty remarkable. So as mentioned, diagnostic is really important, both in the prevention, but also in the detection of acute disease, as we've seen for SARS-CoV-2, but also after therapy. And so we also have a project on therapeutic drug monitoring, which will become also relevant for the Chemo Maker. We have patented a methodology which really allowed us to do this massive amount of tests. So we were doing, 3 people were doing 1,000 tests per day. We did 200,000 COVID tests. We reached a sensitivity of 98.7. We also published a paper. Now we sell this solution not only for saliva, which of course it's our favorite matrix, but also for anything which is a biomaterial also looking into the food industry, which is also increasingly using the same technology as BioTech. And of course, we developed also a proprietary testing model that allows complete anonymization, and also through the barcoding and QR coding. And in this endeavor, we're also collaborating with an industry, co-medical in Trento, with the help, of course, of GN Life and BioValley, to really export our conservation product and our method also beyond Switzerland. And so in this journey, of course, we now, after the pandemic was finished, it was officially finished in March in Switzerland, we had to think about new products and how to use saliva for something that we have very at core in the prevention. And so, of course, prevention seems something that was far away. We all, we live a long life, and so we have the opportunity for preventative medicine. Fact is that at present, about 60% of diseases are chronic. There is a major impact in society. $7.8 trillion are spent in chronic diseases, and the well-being prevention market has really a margin for increase much more than therapeutics at this time. Of course, the salivary diagnostic in this perspective is very interesting because it's extremely accessible, it's self-sampleable, and can give you a result pretty quickly. And it is expected to reach from 5.7 trillion already, including in COVID diagnostic, an increase in 11% of growth. So in this perspective of salivary diagnostics, we are basically looking at salivary hormone, which we'll start to sell next week, with the collaboration with Tecan, which is a Swiss company, and IBL International, which is a German company. And then we have developed a 16 biomarkers profile in saliva that allows us to identify all area of deficits that will then be complemented with supplement. And this is a business unit of SIC that will then be spun off as a startup. But of course, we have very much a core other project. So we have a periodontitis diagnostic project. We're developing a point of care for a multiplex biomarkers, so 2 biomarkers from pathogen, 1 biomarker from the host, and then my old love, which is of course looking for diagnostic and therapeutics for Alzheimer, which is continuing looking into oral microbiome and host interaction in the mouth in order to detect early on, as soon as possible, changes in immunity and cardiovascular to really prevent Alzheimer's disease. And all it's possible so through the use of artificial intelligence for this big data that we are collecting every day, that then can make a selection of the biomarkers. And this is just the last product that we are selling. I think we have made big strides, because when we got the chemo maker twice in Switzerland and we presented, there were always, Swiss people are particularly good at finding loops and we just fill them. So now we are in good relationship with Becton and Dickinson, and we're trying to integrate chemo maker with Cato from Becton and Dickinson, and this will give us access to all the DACH, so it's Deutschland, Österreich, and Schweiz, so the whole region, to German speaking region of Europe. And of course, we are also, thanks to the collaboration with Giovanni Loser and BioValley, we will try to embed the chemo maker into a BioAir installator, which will give us access to the French-speaking areas. And I think I would like to thank everyone, and for me it's a pleasure to be here for the second year. I think I have to thank my team, because without my team nothing is possible, and we are a very small team but very strong, and we are very resilient.
Thank you very much, Lavinia. It's really amazing work for 5 people.
Yes.
So do we have some question from the floor? Alessandro?
Yeah.
One aspect of your approach is that you have really you can process around 1,000 samples per day. Okay,
Yeah.
this is impressive. However, the other application that you are planning are low throughput in a way. I mean,
I think,
cannot imagine 1,000 tests for
Wellness.
Alzheimer.
no. As a matter of fact, so we operate in a very opportunistic way. So we do services on one side that with high throughput and which requires automatization, and on the other side we do research. So basically we do the service with very large throughput. Yeah,
It's fine. What I was thinking, if you ever considered multiplex kind of approaches for symptom-based diseases, for example, respiratory diseases, you can have a panel of different pathogens and you can test them at the same time. And in that case, the high throughput that you have developed would make a lot of sense in this case.
But I think, probably we are not ready to hear the news right now because we have no therapy for Alzheimer's. But I think the diagnostic for the future, and we know the brain ages as soon as we are born, right? So the testing, I think, in order to implement preventive strategies or therapeutic strategy, or to do segmentation and get into a clinical trial, will start at 40. And I think that everyone would like to get a profile, which is of course robust enough in order to predict your clinical trajectory. So I think that in that sense, high throughput will make a lot of sense. I don't think we are prepared right now because we're really bothered by a lot of the failure in this industry. I can think about a lot of opportunity for therapy, especially looking at the interaction between immunity and vasculature. Yeah.
Thank you very much.
Lavinia, I was just going to piggyback on Alessandro's question because I know my years with working with Sue Heldman and the Gates Foundation, they were looking for inexpensive ways to identify the big 5 viral problems in Africa and some other places. And very impressed, obviously, with the work you're doing. And as someone who has a mother with Alzheimer's, I'm hopeful that becomes something that becomes standard. But I think the idea of a saliva test in Africa or in small emerging countries is not a problem. Sounds like you've conquered the saliva testing challenge. So.
That's what they say about us. But of course, we need also to bring in money, and so we go into the wellness industry as a preventative approach, and of course then continue our endeavors into the research where we have very, very strong evidence for this interaction between host pathogen interaction that then leads to a vascular type of dementia, that it's a mixed AD. So we are operating there, but we're making much smaller steps and much more accurate because, of course, also the field is very old style. In the U.S., though, I think your investors would be happy to know that there's a $20 billion unregulated wellness market. So. I know. Yes, thank you.
Thank you very much, Lavinia. So we call the next speaker. It's Francesca Spazzali. She's a product specialist and marketing assistant in GN Life that belongs to BioValley Group. The floor is yours.
Buon pomeriggio a tutti. Grazie dell'invito. Nel mio intervento di oggi vi voglio brevemente illustrare di come il Covid sia stata un'opportunità per lo sviluppo di nuove
on how COVID, SARS-CoV-2 became an opportunity for the development of new technologies.
Sfide rappresentate sicuramente da un sovraccarico dei laboratori stessi per il gran numero di
These challenges were due to an overburden of the labs because of the assays and analysis they had to take, with lack of material and personnel and extraction times that were quite long.
risolvere tutte queste problematiche. E un esempio virtuoso in questo senso deriva
A virtuous example is the the case that we just saw, following a campaign, a mass testing campaign, a new saliva molecular test was created for SARS-CoV-2. The method was devised and patented by SICK, and it works on a native saliva, and it is based on a heating protocol which allows for a rapid liberation of nucleic acids without the extraction and purification steps that require a longer time, and also a lot of materials and reagents. The method was validated through a clinical study that highlighted that the specificity and sensitivity ratio are very high and comparable with the golden standard. So the method is totally reliable. There are advantages, of course, and they were clearly visible. It allowed for a 3 times faster and 5 times more efficient analysis for the extraction of RNA. It allowed for the preparation of the sample of PCR using the equipment already available in the lab. So it didn't require any external equipment. It was successfully used in Switzerland, where a lot of tests were carried out, and this confirms its reliability. So we were wondered if this technology could be enlarged to other applications other than COVID, and this is how the Mixiera was born, a lysis buffer created for the liberation of the nucleic acid solution. Mixiera was the protagonist of the EuroBioHighTech in May, where we discussed it, and we saw that it was 3 times cheaper, 3 times faster, and it is completely a plug and play mechanism, and it is also available for other biomatrices, and it is suitable for several applications, not just diagnostics, but for example, foods. It is a field where the results are very promising. We had stepped forwards in the past few months. There are a few labs that are using it, and there is some inputs that tell us that we can create even more functional and innovative products. As we saw in the previous presentation, saliva testing diagnostics will be increasingly used in the future, even because it is easier to collect the sample. It's less invasive and easier than collecting a blood sample. There are many application fields where saliva can be used. As you can see a summary in the table. So the main ones are infectious diseases. Saliva is very used, and it will be increasingly so, but also wellness and precision medicine will see the increased use of saliva. So thanks to the cooperation with SICK, but other partner companies, we are working on other products that are working this way and can be used for saliva testing. With SICK, we are working on 2 new solutions, Xera 1 and Xera 2, and together with CoMedical, we are developing a device for the collection and preservation of saliva. In particular, Xera 1 and 2 were devised also following the suggestions that we received over the past few months in our dialogue with several analysis centers and labs. To put it in easier terms, We are using the stabilizing buffer to preserve saliva at room temperature for up to 4 weeks, which is compatible with all extraction methods. And then we have a lysis buffer, second step, which is a rapid and economic processing of the sample without any interference with the subsequent analysis. The second productions being developed is a kit for the collection of saliva. It is an upgrade of the already excellent device designed by Comedica, to which we would like to add particular features. For example, the buffer and stabilizing part, which will allow the device to be used in various fields. One of the priorities is the addition of a preset collector, pre-mounted collector that facilitates the collection of the sample. The collector is not thrown away, and it is a potentially infectious material, so you don't throw it away in the waste. And then the last addition is a QR barcode on the tube and a QR code at the bottom, which will allow the easy registration of the operator of the device. It will make easier the acceptance of the sample in the lab. There will be this QR code at the bottom, which will make things easier to register the sample and then process the subsequent analysis. So by adding these characteristics to the device, we will get a very competitive device versus the devices currently available on the market. If you have a look at the main features, we are in an excellent position and we are highly competitive in terms of the presence of the stabilizing buffer and very low contamination risk, because the collector stays in the device. It's a very easy and safe collection of the sample. It can be independently, and it will be compatible by adding the barcode and the QR code with robotic systems, so it will be available for automation in the future. So to identify briefly the solutions available for the market, Xterra and the device are available for analysis lab, clinical labs, hospitals, pediatric, senior homes, where it is needed to collect samples in an easy way, also because there are vulnerable patients. And then we have all the sector of wellness and then academic labs. I'd like to conclude with the last slide, in which I presented to summarize the possible potential clients, hospitals, operators, and the community, and the main problems that each cluster can solve, and then how our products can offer a solution. So a device that is not invasive, that allows for easy collection of samples, and how this is transferred to the hospital, where it won't be needed to employ staff risking infections or even avoiding an overload of patients. We saw that in pharmacies recently, personnel exposed to possible infections. And then, as concerns the labs, our solutions can guarantee an easier management of the sample and an easier and reliable analysis of the sample.
Thank you very much, Francesca. It's really nice presentation, and I have to say that it's really pleasure to see over these 2 years the progress, very focused progress for such a small company. I mean, you are doing amazing job. So do we have any question or comment?
I just applaud the portability and the focus on portability, giving the need to democratize care into parts of the world that you have to transport these samples out of, and the temperatures are not always conducive for those samples to save or to be relevant when they get there. So congratulations on some great work.
Thank you. Thank you very much.
Yes, we have one brief question.
Può ripetere, scusi, la domanda? In quanto tempo?
How many samples can you analyze in 1 hour in one day? With the Xiara method, the experts will be able to provide more details, but potentially, with the traditional method, extraction method, a sample requires an hour. In this case, it requires 5 or 20 minutes, 5 to 20 minutes. With foods, the analysis carried out on milk and yogurts, it only required 5 minutes. The usual kits require 180. So the time is really halved, more than halved.
Thank you very much. We can go ahead. So I would like to invite our next speaker, Kevin Einger. He is the Biotechnology Development Manager from Seric in Terviscosa. Kevin, please, the floor is yours.
So, good afternoon. It's nice to be back again this year. It's nice to see people with faces again. It was quite a while. At least that way I can see a smile, a smirk, a frown. It helps a lot when you're talking. Today I'd like to introduce Serikim to you, talk about the major project we're working on, give you a little bit of a perspective about biotechnology, and then give you an example of the synergy between the biotechnology and chemistry at Serikim. Down? Okay. So Serikim has been an established fine chemicals process development, analytics, and small-scale production company for almost 20 years now. So we would be defined as a CRDMO, and it's a bit like alphabet soup, but it reflects a lot of experience in fine chemistry and the opportunity to fill the needs of a wide variety of clients. So a lot of experience and a wide variety of clients. The old foxes in business will hear high fixed cost and unstable market. Okay, so I'll talk about the collaboration we've been doing. Natasha at the ICGB, technology transfer for biosimilars. Biosimilars, as I'm sure you all know, are the generics of the biologics molecules. All right. So a generic or a copy, a biosimilar of an existing biologic wouldn't seem to require innovation, making a copy. But there's a tremendous amount of innovation involved in this process. A drug that's been licensed, been sold for more than 20 years, and then has gone out of patent, represents also very old technology. Industries, the industrial cost for retrofitting is so large in the pharmaceutical business that these processes for drugs aren't changed. They're very static processes. The challenge for producers of biosimilars is to upgrade the technology, introduce innovation. Maurizio was saying Pharma 4.0 is old hat, but that's what we're doing, old-fashioned science, I guess, Pharma 4.0. And So that's our goal for the biosimilars. The principal goal, of course, is maintaining quality and reducing the cost of goods. I would point out that cost of goods, especially if you go outside the very, very large markets, doesn't really represent the cost per patient, but the cost of goods represents the number of patients that can be treated with a finite amount of resource. So that's what we've been working on. Okay, so if I have the, I can do this? Yes, perhaps. All right, so the major project is the insulin biosimilars. Insulin was the first generic molecule 100 years ago. And since then, the costs have continued to increase. The need is ever greater, and the lack of insulin has been increasing year by year. All right, what we've been doing is taking the strain, improving the yeast strain that is being used to produce it, doing the process development for insulin itself, and two long-lasting once-a-day versions, right, at a competitive cost of goods, as I mentioned before. The things that I always focused on, and we were really fighting hard, are this property rights for our production, process optimization and automation, helping the producers produce their own local supply of insulin. So we're doing inshoring support. And as we develop high quality reagents, we want to provide these reagents to our clients. I'd like to mention a little bit here a perspective about biotechnology. Biotechnology in general is when we take a biomass, which is a high energy material, and we transform it into a lower energy material. We do that, and we either extract the molecules of interest or energy, or both. I think it was interesting to say just to mention, Dr. Banks mentioned that this biotechnology can either be high technology or low technology. Low technology has much wider application. The head of the ICTP research, I mentioned this morning that the creation of this new institute years ago created a bit of this ecosystem, and he referred to it as creating humus or compost. So I think that Diego's model for an ecosystem actually should be modeled on a decomposition model for an ecosystem. In order to sort of give you a perspective, what does it mean to be a biotechnologist? I'd like to explain this by an example. So, two biotechnologists meet in the bar. This is something that, if you know biotechnologists, shouldn't surprise you. And what is it of fundamental importance to a biotechnologist? What would I want to know from another biotechnologist? And there are just two things. What biomass are you working with in quantities? And what is your microorganism? And that's the basis of all the biotechnology in these forms. We can survey a region, look at its biomass, and then put together the microorganism which is appropriate. In a more technological atmosphere, what Natasha is doing with the mammalian cell cultures, or what we're doing in Pichia, in Sericum with the Pichia yeast, is putting together the right biomass and the right microorganism. The success story that we have in Sericum, putting a biotechnology company department in a fine chemical company has many advantages. One is that all the analytics that we need are provided by our colleagues, which are chemists. And the other advantage is that we've been able to do an example of biocatalysis. We have developed the process for miglustat, which Marcello mentioned. Miglustat and the related immunosugars are used for the treatment of lysosomal storage diseases, among other things, for the non-viral application. You can see the structure of miglustat. It's a very simple sugar, but it does have 4 chiral centers. Those of you who remember your chemistry will realize that a chiral center is a left or right-handed tetrahedral. And these are important for the activity of this sugar. If we do this in a traditional chemical way, which we add a lot of heat and solvents, these chiral centers become racemized or randomized. All right, so for theoretically, if I have 4 chiral centers, 1/16 of those molecules will be the one I want. We use bacterial whole cell catalyst to do a crucial conversion. It's enzymatic and therefore stereospecific. We convert the sugar into exactly the sugar that we want quickly at low temperature, and also we use a very strange solvent. Our chemists are very, very shocked at our use of this very, very bizarre solvent, and we call it water. They have a lot of difficulty working with water, but we're getting some progress. This is also very important in terms of cost reduction and also waste reduction. We have a lot of waste reduction. We have a lot of costs with our waste here in the European Union, and therefore having a process which is well developed from beginning to end, and also for the disposal is critical. Thank you.
Thank you very much, Kevin, for your presentation and work done. So do we have some question or comment?
Kevin, I just have a important question, but really it's as well as a comment. As we think about the cost of healthcare increasing significantly, obviously in the U.S., but globally, the GDP, pressure on GDP today, given the world economy, and that's not going to get better probably in the next few years, still left over from the pandemic. Are you seeing more interest in sort of the generics, as we call them, or the biosimilars, outside of just the world of insulin and diabetes? And specifically, where do you think we can go in oncology? Because it's an incredibly expensive place to work in terms of a physician trying to manage a patient, and obviously there's a big need there. So
I know in general there's huge investments in the biosimilars in the world right now. It's really the fastest growing sector of the business. Some of the high-profile biologics that are coming out are tremendous and tremendously expensive. So I think the ability to distribute these resources as in forms of biosimilars for countries where there was one African country which was spending a few years ago half their health budget on insulin. Yeah. So I think in those situations when the health system is so stressed, oncology is not a priority. Right. So I think if we can actually introduce that as make it a priority, because it's no longer a death sentence. Right. And I think would be a major transition where people have higher needs.
Yeah. Thank you.
And if I can add just a comment, like, yes, if you look in all the graphics or the new drugs developed, biological drugs developed, the cancer is the main part of all these graphs. So
the problem is the U.S. pharmaceutical companies are very proud of those drugs, and they tend to charge a lot for them. And so as someone trying to see democratization of high academic care, it makes it challenging, yeah? So.
Yeah. Yeah, but they have 20 years to make all the money they want, and after the 20 years, then when patent expire, then they can complain. Yeah, no,
Well, there's one thing that most people don't realize is a lot of these cell lines for the production, or in the case of yeast, it's not intellectual property, right? But these are property rights, and these never expire. Right. So the yeast system for the production of recombinant protein has been in control of a single company, a monopoly, for the last 70 years. Yeah. And now the new cells for the production of biologics are also under property right control. So this is a way of increasing the prices of drugs beyond the reach of countries that have less economic power.
Yeah.
Okay, thank you very much. I would like to invite the next speaker. It's Elisa Margotti from Protein Science Department of the company Halifax here in Trieste. Please, Elisa.
Good afternoon. Thank you for inviting me here again this year. So today I'm going to tell you about a turning that has taken place and is still taking place in Halifax Research and Development, a branch of Halifax SRL, that is an Italian company with almost 35 years of experience in the field of medical devices for in vitro diagnostics, that is settled in Padova, close to Padova, while Halifax Research and Development was established in 2017, so only 5 years ago, in the Area Science Park in campus of Basovizza, as a branch of Halifax Holding entirely dedicated to molecular biology diagnostics. This very specialized and focused mission of Halifax R&D changed a little bit, or was somehow upgraded in 2020, when Halifax R&D acquired a facility that was already present in Area Science Park and was destined basically to this mission and oblivion. And it was a facility fully equipped to follow the whole process of selection and screening of monoclonal antibodies and production and characterization of recombinant proteins. And together with this facility, Halifax R&D also acquired a team of trained people who brought to the company all the skills and expertise to design, develop, produce and deeply characterize proteins of high quality and competitive costs to improve Halifax core business in the market of diagnostic tools. And in less than 2 years, since 2020, since the moment of acquisition, the Protein Science Group has already achieved very important results. That's to say, the production of 2 proteins as raw material for diagnostics tests that have been implemented in, and commercialized already by Halifax. And this process have been followed for the very beginning, from the start. So designing the more appropriate constructs for these 2 proteins, the constructs that have been optimized also for expression XP293F mammalian cell lines that have been chosen because they guarantee a very high yield of expression. From the supernatant of these proteins have then been purified, setting up the most suitable protocol for each specific protein. Here in the picture, I show you the results and the very good purity that we can obtain. We have obtained already after 1 single step of purification, so after the immobilized metal affinity chromatography. As you can see, the diffractions of the chromatography already give a quite good purity level that basically becomes almost 100% when the purification is then concluded, and we pass to the characterization step. So we can we apply different techniques, for example, SDS page and Western blot to check protein integrity and purity. And as you can see from SDS page, our protein really has really good level of purity. But of course, with an SDS page, we don't have enough sensitivity to check for small amounts of fragments. But with Western blot, of course, we have an assurance that is better about the purity of our proteins. Then here, I can show the 2 chromatographic steps, 2 chromatographic peaks to check oligomeric state of our proteins. As you can see, our proteins elute in 1 single peak, again meaning that our proteins are in a monomeric state. We don't have aggregation or fragmentation of our proteins. And in particular, in this picture, I show you the overlapping of the peaks of the same protein, but 2 different purifications, so 2 different batches of our protein. And as you can see, the overlapping is really perfect. So we have also good consistency between different purification performed in different moments. Then, as these proteins, and I can tell you, are the receptor binding domain of SARS-CoV-2 spike protein and its receptor, so angiotensin converting enzyme 2. So as these two proteins are supposed to interact, we also had settled up a test to check the kinetics of binding through Biacore X100 instrument. Here you can see the kinetics of binding of the two proteins. So AC2 is captured as a ligand, and then different concentrations of spike RBD are applied as analyte. And as you can see, the different curves only depend on the concentration of RBD. And the kinetic constant is around 20-30 nanomolar, and it's in agreement with what you can find in the literature as referring to these two interactors. So we have also good correspondence with the literature. And then in the end, as ACE2 is an enzyme, as the name says, we also set up an enzymatic activity assay to verify that our protein, after the purification and all the steps of purification, still keeps the enzymatic activity. Here in this graph, you can see how the protein purified in Halifax in comparison with the commercial one, and the slopes of the two lines are basically the same. So it's the cleavage activity of the peptide. Thank you. The cleavage activity of the peptide we have used as substrate is the same for our protein, comparable with the commercial one. After all this work, finally, the two proteins have been implemented in one rapid test commercialized by Halifax. And this has concerned the part of production of recombinant protein. Now, let me just tell you something also about the field of selection of monoclonal antibody. At present, we are in the process of acquisition of a human naive antibody library to set up a screening approach for identification of the best antibodies against a pilot target that we have already selected. And after this first step, of course, in the future, we are going to define further key targets for library screening in order to improve the development of diagnostic and prognostic tests implemented and commercialized by Halifax in order to meet at the best the needs of the market. And then, of course, we hope to be able to set up a proprietary library to broaden even more all the possible application and to enhance and boost the know-how and the value of the company. In the end, of course, I really want to thank all the people that have worked with me, so the other people of the protein science team, and especially Yvette for designing and also setting up the purification protocol, Marco, who basically set up all the binding protocols and also the enzymatic activity assay, and Letizia, who's of course helped us at the beginning in the expression of this protein, but now is the person who will be mainly involved in the process of acquisition of the library and then the library screening. I, of course, I really want to thank also all the molecular mouse team and the molecular biology people, because the atmosphere that they create in our lab is always friendly and stimulating. And last but not least, Martina, who's in the office, and he takes care of all the administrative stuff, and we can concentrate on research because she takes care of all the rest. And thank you very much for your attention. Hello.
Had a new breath of air in these last 2 years and obtained such great results. I had just one question. So you're using transient transfection.
Yes.
So how do you standardize the production batches? Just so is it challenging task?
Well, I have to say that for the moment, the system we chose, so these XP293 cells, are quite reproducible from one transfection to the other one. And thanks to their really high yield of expression, basically we have done a couple of transfections and we obtained really a huge amount of proteins. And we checked the concentration of the protein in the supernatant between one transfection and the other one, and this is quite reproducible. So, and also, for example, picking the same volume of the supernatant and performing a Western blot, for example, to check that our target is there with the tag that we use for purification, we more or less obtain the same kind of results at the end. So for the moment, let's say that cells are quite good in this. So they are reproducible as a system, as an expression system.
Okay. Thank you very much. Alessandro?
For the purpose of the RPD, the assay that you are doing is a neutralization assay, right? The assay that you are doing, RBD plus AC2, is an antibody neutralization assay that you're running in your device. Is it?
Yes, we are talking about
the final...
the enzymatic or the BIAC or the binding?
No, the final device, the final antigen test that you are showing.
Okay.
It's a neutralization.
Okay, sorry. So the final test is assembled in NIMIS.
Yes, but it's an antibody neutralization assay.
Yes.
Okay, so it depends on the kind of RBD, because there's a lot of mutations of RBD in COVID, right?
Yeah.
So it changes with the different viruses that appear. So it's good to have a strategy that you don't have to prepare a cell line for each of them. But if you can transfect and rapidly purify a variant, you have an advantage over...
Sure, this for sure.
Yeah.
No, this for sure. I mean, of course, at the beginning, we started with transient transfection because there was this hurry of having the product, of course. And again, as the system is so efficient, we could basically produce one huge lot of final tests with just one transfection. So, basically, yes, of course, this was the reason.
Of course, that is the main advantage of transient transaction, but the consistency of batch, usually where regulators or whatever, they object.
No, the reason why we have so many final tests and checks for the proteins, because everything has to go into its place, and being sure that our batches are consistent one to each other.
Yeah.
Thank you very much.
Maybe we have quick...
Turnaround time from transfection to the protein.
Okay, let's put it in this way. If you go straight from transfection to final protein, 1 month. But being the supernatant so rich in protein, we could just store part of the supernatant taken from cell culture at -20, and after that purification, then it's a couple of weeks. And I mean purification and all the tests after purification to be sure that I have to check the batch, and it's a couple of weeks.
Okay, thank you.
Okay, thank you very much, Lisa. So I would like to invite the next speaker, Mikol Kozi Sadok, or Sadok, so I'm sorry if I didn't pronounce your surname properly. Sadok? Yeah. Product and marketing manager from GN Life, here from Trieste, please.
Grazie, io sono Mikol Kozi Sadok.
Thank you. My name is Mikol Kozi Sadok. I work for GN Life, which is an enterprise of the High Valley Group. Our mission has always been the same and has never changed in time, even though the company and its proposals matured over time. That is, to improve the quality of life of people, which everybody declares, but what distinguishes us from the others is that we try to do so thanks to the studies on human genome applied to well-being. Our target audience is the final consumer, the end consumer. So people who don't know the sector, who have no skills in the sector. The company was founded in 2009. Up to now, many people know what a saliva swab is, what PCR is, and what COVID tests are, which is a relief in some respects, because already in 2009 we did use these devices, these tools, to offer our services. In 2009, we were among the pioneers in this field. If we think that human DNA sequencing at the beginning of the years 2000 needed a huge investment in terms of money and time, over the last 20 years, much has changed, and our progress enabled a much wider usability of the technology that can also be applied not only to diagnostic or medical fields, but also to prevention. Today, even more compared to 2009, we know much more about the relation between genes and the environment, because we know that the environment or the ingredients or specific circumstances impact genetic expression. We know this today thanks to our technologies, real-time PCR, sequencing, microarray, that permit to see how some ingredients activate or deactivate some genes. The coronavirus, as we've already seen today in the previous presentations, enabled a huge technological advancement, both in the analysis methods in terms of types and quickness, and in the development of saliva diagnostics, which also permits new approaches to human well-being, because the paradigm changes. It is the body that drives the choice, thanks to the discovery in the field of genetics and epigenetics, and thanks to the new technologies. What are the core pillars, the core focus pillars of the company? Nutrition, skincare, and sport. We know that the NRRP allocated much financing to these projects, but with the aging of the population, which is increasing, and a longer life expectancy, it is crucial to consider prevention from chronic diseases that occur starting from 50 years of age, approximately. So we concentrate on three fields: nutrition, sport, and skincare, as you can see on the slide, that can be gathered within a larger field that is longevity. I'm not talking about 100-year-old people, but how to stay younger longer. How do we do this? With two sides of the same project. That is, on the one hand, we do propose programs, genetic programs, through genetic analysis. We look at the peculiarities of the individual to offer nutrition services, sports services, and on the other side, products. And I mean nutraceuticals and cosmetics, epigenetic products, because we can see more and more how ingredients impact genetic expression. As I said, we've been among the first to offer this type of services, so we had the chance to have a label in the field of nutrition, combining individual genetics and the health status of the individual. We carried out a study that lasted 2 years to see how to consider genetics and the peculiar traits of an individual within a nutritional program can help us obtain better results and can help us maintain the weight achieved through the program. And this effect can also be translated towards the other areas we concentrate on it with our projects. This type of programs moved from a DNA analysis, which we all know in our daily lives, and which up to 2009 was an obstacle to communicate to the individuals, to the people who decided to entrust us with the analysis. So thinking of a DNA aside through saliva testing was pioneering, whereas today it is our daily routine. On the other hand, I was saying we have products. These products are not new, but they had a giant leap forward thanks to coronavirus because since 2020, we should concentrate on the pandemics, and we had to concentrate on the other side of our product, which is what can we insert in our bodies so that our bodies can react. So the process is always the same. Identifying a problem, for example, stress, identifying one or two or more epigenetic ingredients that can positively regulate our genes, our genetic response, and finally, a biotech treatment, thanks to local technologies developed in Friuli Venezia Giulia to modify chemical and physical characteristics to obtain the best results possible. One example, rhodiola rosea, which is an extract, an ingredient widely used to combat stress. It can positively regulate over 1,000 genes, some of them linked to the management of anxiety, concentration, and behavioral traits in general. So was the first ingredient to use, and thanks to the technology we applied, we managed together with our partner to create an ingredient, RhodioX, that is 10 times more active on the production of cell energy, ATP, compared to a traditional extract. GN Life today has two main branches, the B2C one, that is targeted at end consumers, thanks to our brand, Generame. Our products and programs can be purchased directly online, and a branch of the company is the B2B one. We are a collector of biotechnological know-how and skills to bring value to end consumers and to other companies. We've been collaborating with different institutions over the years, not necessarily linked to the genetic or epigenetic world. We work with a multinational company whose core business is the fight against air pollution, for which we've created products and we keep on producing services. We also have an ongoing collaboration to identify saliva biomarkers and to propose associated products. And to conclude, what we do is to try and bring innovation down to earth, to have hands-on on our know-how and skills in our region, to bring added value to the different players, be them the consumers and the other companies. Thank you.
Thank you very much, Nicole. There's very nice presentation. Does anyone have any question?
This is an area of great interest for me personally, but because I'm old and have seen the movement from PCR through full-scale genomics and now into RNA-seq and ultimately back to the transcriptome and epigenomics. In 2018-ish, the U.S. government became scared and overreacted to consumer genomics and allowing people to easily get their their epigenomics done and discouraged it. And so we were in the middle of looking at large-scale studies to look at the impact of epigenomics in things as you're talking. And it seems like you're not seeing that issue in Europe, which I think is true, but because of the pandemic, I haven't been over in a while. But it seems like you're getting access. Are you just still using big GWAS studies and using that data, or are you actually seeing data from consumers?
Well, it depends. For the programs, we analyze we get analyzed the actual DNA of the consumer,
right,
who of course has to sign up privacy, and everything is locked down together with some consultants that took care of that aspect. For the ingredients, we are mainly based on by blog people, international studies, and what's already established.
But consumer sentiment in Europe is positive towards epigenomics and giving a sample?
I think that COVID makes people more acquaintance with this subject.
Yeah.
Of course, in the US, you are forward because everything started with 23andMe and the main players, and we are still some step back. Behind, yes. But I think that the importance of prevention and on the other side, to understand that the players in the sectors are here to help, has been boosted by COVID. So I think that the final consumer now, it's more open to this kind of technologies and possibilities.
Yeah, we are seeing that in the U.S. as well, which, as you know, is a huge opportunity for wellness products. So, fascinating work. Thank you.
Thank you.
Thank you very much. Okay, we can go further, and I would like to invite our next speaker, Daniele Gulic, CEO from Sky Proxima company from Trieste. Hello.
Buongiorno a tutti, io sono Daniele.
Good afternoon. My name is Daniele Gulic. I am one of the founders of Sky Proxima, a startup specializing in aerospace technology. So what does aerospace has to do in this context? We are operating in an innovative context, but a very fast innovation and evolution. In the next 5 years, it deals with advanced air mobility, and we are adapting the concept to air mobility, to the healthcare. What does it mean? We developed a program called Filotea, which will take to the Italian territory, or we will be the first European company that will bring to Italy a platform for healthcare transport with aircrafts, electric or hybrid aircrafts, but also accompanied by operational facilities, new logistic models, and most importantly, the thermoregulated box that will be integrated within our aircrafts. We've done everything on our own. We've created exclusive partnership with companies, for example, for the box, a thermoregulated box, which is patented at the European level, and it is integrated, built in our aircrafts. In the program this week, we have brought into the program a British company that created the first world's operational drone airport, UAP UK. We signed an exclusive cooperation agreement to bring it in our program. So in the next year, the first 6 months, in the Gorizia airport, there will be an airport dedicated to electric aircraft with vertical takeoff. which feature an integrated system for the logistics of medicines and blood transportation. And it will be a management and control system that will serve the first cross-border routes, for example, Italy and Slovenia, and Italy and Austria. So when I'm talking about drones, you don't have to think about the small drones that you see, but these are aircrafts that are able to fly between 80 and 150 kilometers, and they take off and they land vertically, and they work basically as an aircraft, a traditional aircraft. They can reach far away places, and they are able to tackle emergency situations. For example, the transport of urgent medicines in a very short time, also reaching places that are normally difficult to reach. In the Filotea program has already been certified C-block by the Register of Biologists and Pharmacists. And so we are the first European program. We have built a healthcare protocol for the airborne transport with means and infrastructure that we are going to present shortly at the national level. We are currently waiting for the framework agreement with the regional authority and the national civil aviation body. And we will be the first region in the country with operations in the field of advanced mobility in the healthcare field. This is not an experiment. The requirements today are for a real service. So we need to implement a service for analysis labs, and we need to be able to provide first aid in the most diverse situation. This is my only slide, but it exemplifies all the elements that are included in the program. We're talking about a project that was not existing. We have engineers in our staff that are planning the operations for this new transport method. We have aircrafts that can work in a urban context as well as the extra-urban context. We have the refrigerated built-in box. We have the operational logistic model. There will be aerial corridors devoted to this kind of transport. There will be new air motorways to be able to manage healthcare transport. This is not a utopia. 7 years ago in Great Britain, we already saw the first air highway in this sector. And Great Britain is already operating in this context. In America, these type of operations are already active, and the service has been consolidated. And in relation to the US, we were called by Ohio to implement the first service in this context. As I was mentioning, this is a operational model that can be exported. And then the dedicated airport will also have to be able to handle all the elements that are connected with healthcare. This is going to happen soon because within the next the first 6 months of 2023, we will be able to launch the healthcare airport in Gorizia, and it will be the first time in Europe. Ours is an engineering company that creates aircraft, but we have a strong cooperation with important partners that allowed us to create all the infrastructures. And today, one of the most sensitive issues is drones. So the problem is to guarantee the biological integrity and to create the infrastructure to connect hospitals and other laboratories and other facilities. We are working in the aeronautical field. Do you ask to try a Boeing before using it? Well, no. So we have to make sure that our aircrafts work properly, and we're going to do so through a control room located in Gorizia, but they will be able to work around Europe, Paris, Germany, maybe even Moscow, who knows, in the future. And so there will be a control room which will allow us to safely manage the operations of the aircraft from our headquarters. And I can tell you that we're already working. We're already working with SpaceX by Elon Musk to plan the first experiment, the first test to use the satellite network to put our aircraft network in connection. And so we want to create a global network. And this is a very ambitious project that we have. So thank you for your attention.
Thank you very much, Daniela, and I'm really happy to hear that since March, when we heard your first presentation, that you obtained this really important partnership with UK partners. So I congratulate you.
Yeah, this is a very important partnership because we are really ready to put on the land a real infrastructure. It's important for us to create a network and a hub everywhere, just to link cities, hospitals, and laboratories.
I can have just one question. So for the developing countries, how do you see this? Like, is it something that it's really sustainable, economically viable in the remote regions, and maybe delivering the diagnostic kits, like we heard the guys who are developing them? So in the
for example, we are now working and we are speaking with the African country, in which they are very important to establish specific activities like this, because as you know, the distances are very high, and sometimes there are no possibilities to connect because the field is not permissive for car or other infrastructure or other type of transportation.
But also, if I can say, like talking on some other reunions with the people who work in African countries, they said that the main problem have that people do not go to the hospital, but the hospital have, or the drug or the diagnosis have to come to them. Yeah. And that's really big challenge. So
this is the challenge, but not only for African country, also for European country, and also for Italy.
Yeah. I think Lavinia has...
Yeah, I have two questions. One is, who's controlling the airway for drones? And second question, are you paving the way for passenger e-voting?
In this moment, ENAC, that is the institutional government for the rule of flight control, is run up to give us the opportunity to create this highway in this sector.
We are working to flight on 200 meters, for example, and probably we will have the exclusivity to flight on that area only for medical sector. And in the first 3 months of the 2024, we present the first drone for transport, for passenger transport. Yes.
Because I think in Dubai there is already a project on that.
Yeah, Dubai already is, but at the moment there are a lot of industries that are working on. Yeah. But for example, they have developed specific aircraft that are capable to fly for maximum 15 or 20 minutes. The challenge is to create the new propulsion mixing electrical and hydrogen technology.
Yeah. Thank you.
Okay. Thank you very much. Ronnie, do you want to add something?
I'm in the wow mode. I grew up in a very, very rural area of the United States and can only think about how far we had to drive to a pharmacia to get the meds. And I imagine CVS and Walgreens and the large pharmacy companies in the U.S. will be begging for your service because it'll be a differentiator for them to reach the rural areas. So really fascinating work. So
and this moment, as I told before, the U.S. are has more advantages for this type of technologies because the FAA has already put the rule for this type of flight.
Can I ask one crazy question?
Yeah.
What's the average age of the drone drivers? 20? What's the average age of
the average age? We started to teach from 18 years old. Yeah.
My age, not excited. Yeah.
Because obviously it will be the new work.
Yeah. Right.
A new work for young people.
Yeah.
Yes.
It's exciting.
Yeah.
Okay. Thank you very much. Okay, I would call the next speaker, that is Vincent Torre, BioWireless System and Solution here in Trieste. Please, the floor is yours.
Allora, bene.
First of all, I would like to thank you for inviting me here. We'll introduce BioWireless Systems and Solutions, which is an innovation and innovative startups we founded less than 2 months ago. Currently, the board of directors is quite small: Diego Braver, myself, and Paolo Giribona. That said, we are happy that if we receive more applications with new skills and new resources. For the time being, our project of BioWireless Systems and Solutions works on 3 main axes: automation, and the chemo makers that Lavinia already talked about, treatment, and monitoring. Our main objective is health, of course, and specifically cancer. Another strategic guideline we are interested in is the development of data sets, sanitary health data sets, and I was glad to hear President Federica this morning say that the region was happy to make data they have available. But the problem is not so simple to tackle because of technical and privacy-linked concerns. But having said this, it still is a very important strategic field. Chemomaker, which is the first strategy, is the automated preparation of chemotherapeutics, which is a project that's already ongoing and that BioValley has already developed
commercializzato.
and also marketed.
È qualcosa che rende la
ChemoMaker
preparazione dei
is a device
preparati chemoterapici sicuro,
that
accurato e anche
facilitates the
riproducibile
preparation
e
of
anche permette
chemotherapeutics.
all'operatore di non
It
avere
is
i rischi di poter
reproducible
fare questo
and it
in
curbs the
condizioni,
risks for
diciamo, di
operators, which
sicurezza
means it
per
increases
l'operatore, per il
safety
paziente e per
for the
l'azienda.
operator,
Il
for the
ChemoMaker è
patients,
già in
and
fase
for the
di
users. ChemoMaker
commercializzazione e
is already
è
being
stato già
marketed.
sviluppato il secondo prototipo, che non è più un
We've already
prototipo perché è stato
developed
già
the second
venduto,
prototype, which
ed è qualcosa
no
che
longer is
noi
a
pensiamo che
prototype,
possa
it's already
essere
being sold.
venduto a molti
We
più
think
ospedali in Italia e
that
anche
it
cercare di
could be
poterlo
sold
vendere
to
sia in
much
Europa
more
e forse anche
hospitals in Italy, and we would like to try
fuori
and sell it
dall'Europa e
in
anche in
Europe
America.
and maybe overseas
Il
and in the
secondo
US.
campo di
The second
ricerca,
field we
la seconda
concentrate
strategia,
on
quella che diciamo fa un pochino più capo a me, è
is the
l'idea
one
della
I am
combinazione
more directly
personalizzata di
involved, is
farmaci
to
per il
make a
trattamento di
personalized
patologie
combination
tumorali. E
of
l'idea
drugs for
fondamentale
treating
si
tumors.
basa sulla
The basic
seguente idea.
idea
In genere i trattati, diciamo,
is
i
the
composti
following one.
chemoterapici hanno come
Usually,
target di impedire la duplicazione dei
chemotherapeutics aim
cromosomi
at
e l'idea in un certo senso
impeding the
mia è
duplication of
che
chromosomes,
forse
and
possiamo cercare di
my
avere come
idea is
target
that
non la duplicazione
we
dei
could
cromosomi, ma la
target not the
duplicazione del
duplication of
volume.
chromosomes, but
Perché
the
quando tu hai
duplication of
lo
volume.
sviluppo di un cancro, non solo devi
Because when
raddoppiare il cromosoma,
a
ma devi
tumor
anche raddoppiare il volume.
evolves, you need to double
Allora,
not
su
only
questo
chromosomes, but
argomento
also the
l'idea
volume.
è di usare,
In
di
this field,
fare quello che si dice
the idea
riposizionamento,
is
di
to
usare già farmaci che sono già FDA
reposition,
o EMA approved
that
per un
is,
altro
using
scopo.
drugs that
E
are
su questo
already
BIS,
approved
la
for
nostra
another
startup, ha un
purpose.
brevetto
Our
e
startup,
adesso nella diapositiva
BIS,
successiva
has a patent,
vorrei un pochino far vedere qual è l'idea che sta sopra. Allora,
and this
nel
is the basic
pannello
idea.
indicato prima del trattamento, in rosso
You can see
vedete i
before the
gliomi. I
treatment,
gliomi sono le cellule del
and there
cancro
you can see
principalmente dei
gliomes
tumori del
that are
cervello, e prima del
the brain
trattamento i
cancer
gliomi
cells.
sono
Before the
piatti, hanno
treatment,
molte
these
propaggini, sono in grado, hanno
gliomes
una grande
are
motilità.
flat, they
Dopo il
have many
trattamento
branches
col nostro
and are really
mix, i
mobile. After
gliomi diventano
treatment,
delle pallotte e non
they
sono più in
become
grado
some
di replicare.
smaller and more
E
compact
noi
balls that
pensiamo
cannot
così che
be
questa sia
replicated, that cannot
un'idea non solo
replicate
interessante dal
themselves.
punto di vista scientifico, ma
We
possa
think
essere
this is very interesting,
trasformata
not only
in
from a
un
scientific point of
business.
view, but it could be
L'altra
a
idea
business case, a
che
business
noi
scenario.
abbiamo nella questa startup è la rilevazione della
Another
concentrazione di
idea we have in
farmaci
our startup is
attraverso, diciamo,
identifying
dei
the concentration
bionanossistemi.
of
Per cui la
drugs
nostra
through
idea è quella di cercare di
bio-nanosystems. Our
affrontare il
idea
problema della cura del cancro da diversi
is
punti di
to
vista.
tackle
Con il ChemoMaker,
cancer treatment
l'idea di
problems with
sviluppare
different
nuove
tools, with
combinazioni
ChemoMaker,
di farmaci e il
by developing
riposizionamento,
new
però
combinations of
effettivamente, avendo
drugs and
sentito le
repositioning them.
presentazioni precedenti
Having
di
listened
Lavinia
to
e di Kevin,
the
non
previous
escluderei
presentation by
anche l'idea
Kevin
di,
and
con la
Lavinia, I
collaborazione con
wouldn't
loro,
even
di pensare anche a
exclude with
sviluppare dei
their
farmaci
collaboration
nuovi, più
the idea of
sensibili, con dei
developing
target
new
che
and
poi
more
possiamo
sensitive
discutere insieme.
drugs with
Vedendo che il
targets we could
tempo
discuss together.
stringe, io mi fermerei
I know
qua
the time
e vorrei
allotted is running
ribadire
short, so
che il
I will
progetto
stop
di
here,
mettere
but I
insieme
would like to state
innovazione
once more that bringing
scientifica e
together
tecnologica con business è
scientific,
qualcosa che
technological
credo
innovation
sia
and
molto utile e
business
produttivo, e
opportunities
sentendo un po'
is
tutte
quite
le
useful and
presentazioni
productive.
precedenti
And
penso che possa
judging
essere
from the
una cosa
presentations I've been listening
importante
to
per varie
today, I
ragioni
think could
inutile
be
sottolineare.
very important
Vi
for many
ringrazio.
reasons. Thank you for your attention.
Thank you very much, Vincent. Do we have some question or comment?
We had the privilege of dinner last night, and I thought a lot about our conversation, and I very intrigued as to about the combinatorial effect of putting these therapeutics together and doing that in a format that can be done rapidly and executed on a personalized basis. The problem with personalized medicine, we all know, is we have lots of data, but actualizing it in a patient becomes difficult at times. So I'm curious as to your thoughts about how far away are we?
Well, so my view is that what we are doing is that we have maybe 10 or 20 compounds which are likely to be effective. And if we are able to do a genomic screening, maybe RNA sequencing on the cancer of the tissue of this given patient, we should be able to provide the best possible combination. Yeah. And I think this is not so far away. I mean,
yeah. One of the things we are doing, and we can talk later now, but I didn't know that, we are developing a gene chip, an RNA chip, with the 5,000, we've sequenced all the genome of oncology of cancer, and now we've narrowed it using AI to the 5,000 RNAs that matter. And that can go easily on a chip, and in 5 hours with the 10 nanograms of extracted RNA, we can provide you that result. So we're about 6 months away from that. But that's the importance of combining what we are doing with what you are doing. You know?
Sure. Let's do that.
Yeah.
Right.
Thank you.
It's good to hear that this will result in more collaboration. Thank you very much, Vincent. I would like to now move to the energy high-tech innovation. So it's my pleasure to present our next speaker. It's Massimo Vecchietti. He's the president and the board of directors of Biogas Servizi from Trieste. Please, the floor is yours.
Buongiorno a tutti.
Good afternoon. My name is Massimo Vecchietti. I'll present the operations of our services, of our company. It's a slightly different topic. Our company was founded in 2015. It was an initiative of professionals and researchers from the area of di ricerca. Our activity implies the management of biogas facilities and management of plant-generated energy. So we have activities, we have fundings from investment funds from America, Israel, etc. Our focus lately is biogas and renewable energy, but also biomethane and photovoltaic technology. Over the past few years, we've had a constant growth. Starting from 2015, we had a very limited turnover. Now the turnover is quite high, as you can see. Our skills are diverse. We do management and environmental biotechnology for energy, so engineering, biology, insurance and legal coverage, maintenance and construction management. All these activities are required for the management of an operational facility that produces energy. These are our plants. They are mainly located in Northern Ireland. Our headquarters, Italy. No, our headquarters are located in Trieste. So the photovoltaic development and other renewable energy passes through the search for locations and sites that are suitable. We, of course, we are investing in wind power and the use of agriculture also for the production of energy. It doesn't mean that we take away resources from agriculture. It means that we add something, because agriculture needs energy at this time. try to convert agriculture towards precision agriculture and the use of the energy that is available. Our company is focused on the use of waste biomass, and our aim is to reduce environmental impact and develop the circle of the of energy production. It is accurate management of biomasses, both incoming and outgoing, which can become fertilizers or other things. The last sector, which is very interesting and it is being developing, has been developing, is the biogas plants produce biomethane, but also CO2. It is now a waste gas. In the future plan is to use it together with hydrogen, using photovoltaic technology in synthetic biomethane. These are the developments that we are aiming to. This is not a proprietary technology, but we have cooperations with other companies, for example, in France. Thank you so much for your attention.
Massimo, thank you very much. Yes, we have one question.
Immagino che lei sia un esperto di energia. Cosa pensa dell'agricoltura verticale, cioè praticamente quella fatta di quella specie di armadi in cui uno
What do you think about vertical farming? Those shelves where you use UV lamps to farm plants. Somebody says that it consumes more energy than normally. So it depends also on what you farm, what you grow there. I am an agronomist. I deal with energy. That is definitely a highly technological and capital farming. The crops that are used, that are grown, are high income. So maybe that is sustainable for this type of production. In the Netherlands, you have peppers, chili peppers, and cherry tomatoes with UV lamps, throughout the year, and probably the Dutch are good marketers, so that probably it works. I know that they use greenhouses there. Yes, they use greenhouses, but everything is automated. Like it can be suitable for places like the Arab countries where energy is widely available. In that case, the temperature reaches 50 degrees Celsius. So you need the refrigeration, yes, but they have sun. So with the photovoltaic technology, the cost is almost zero. So you see. In Italy, we have a problem with energy at this time. So I wanted to know what is the projection in terms of solar energy and the creation of energy from high hydrogen. And the costs are high and the time frame is very long, so it doesn't sound so exciting. we still have to work on it. And then also bureaucracy is very is complicating things. So the systems that the current plants are developing now a few hundred megawatts. So the implementation time is a few years. So together with this crisis, I don't know how we can become partially independent in the production of energy. So the production from hydrogen is a little bit is long term, is a long term project, and the basis is to launch a photovoltaic energy production. In Italy, in this moment, maybe my colleague has more information. We have about 12,000 megawatt that needs to be authorized, which represents 20-25% of the Italian production. So this was a big endeavor from all the producers, but this endeavor is not matched by a suitable response from bureaucracy, from the state, from the administration. This is very limiting. Thank you.
Massimo, I was just wondering, in our region, like Friuli Venezia Giulia, in comparison to the other region and in the world, like what is the pace of this energy high-tech innovation research and all these gas services? So where we are in in this race?
Noi in Friuli Venezia Giulia siamo presenti molto poco.
This region, we have very little operations. We are working in the large part of northern Italy. In Friuli Venezia Giulia, we have small plants and small operations. So maybe this is a historical question. We bypassed the Tagliamento River and we already moved to the rest of Italy to develop technology and manage plants and do development. We will have to bridge this gap, of course, and this is a commitment that we are taking on.
Benissimo, grazie.
We wish you luck and good.
Thank you for the support.
Massimo, for the world, please hurry up. We need your work, so yeah.
Hurry up.
Hurry up.
Thank you very much,
Grazie.
Massimo. Grazie. And I'm sorry that we are 10 minutes late, but we have the last speaker. It's Ronnie Andrews who will
I will
tell
go
us.
fast. Actually, I need to, let me get the turn. This is my first time here, and so I wasn't exactly sure exactly what to present. So after hearing some amazing presentations today, I am going to change my presentation and really go with one major slide. So, yeah? Okay, I see it. Yeah, it's coming up. There we go. Yeah, so as I think about today's topics, it was interesting for me, and to summarize, just there was a silver lining in the pandemic, and it was a increase in international cooperation and an acceleration of technologies that can allow us to democratize new technologies and drugs into all regions of the world, which I think is a very, very important quest. Okay, there we go. You know, I took my wife to Greece for her birthday recently, and as we were walking around, I kept seeing this name Hippocrates, and I came across this quote, and I found it very fascinating because I thought he probably said this last year, but actually he said it in 400 BC. And the reality is, life is short, and the act of medicine or the art of medicine is very long, as we all know. The experience, as we know, at least from oncology, is treacherous. But the last part of this struck me, that judgment is flawed. We are now 20 years past the completion of the Human Genome Project, and that clause is still very, very real and relevant. Every day around the world, in my world of oncology, a physician sees a patient, and that patient is unique. It's an N of one. That cancer is different and unique to that patient. And today, 2,422 years after Hippocrates said this, unfortunately, our judgment is still flawed in terms of how we manage disease. This is leading to a really important crisis, economic crisis. And you can see some of these numbers as you look at this slide. But the reality is that we will spend a significant amount of money next year on new therapies and new diagnostics, and yet only 57, this is US data, only 57% of the clinicians actually use the new technologies. So the session before our session, I sat and took notes and was very intrigued with the educational challenge that Italy is facing and how to educate the consumers of this new molecular information. Not only the consumer, the patient itself, but also the physicians who treat the patient, and how rapidly the information changes, and yet it takes a long time for that to become standard of care. And then if you take that and you look at the cost of the drugs that we have created in the United States, and the importance of these drugs in managing these diseases as an N of one, but then you look at the fact that after all of the work that's been done over the last 50 years to develop therapeutics for cancer, today, even today, less than 25% of the drugs that are given to a patient have a durable response, which means 75% of the time a patient gets a drug that will not help them. And unfortunately, the economic impact of that is greater than around the world about 150 to 300 billion dollars in immune therapy alone that will be spent by 2025 that will not impact the patient. And this is a real challenge that we have, and gives one of the reasons I had mentioned this to Diego in our first meeting. I actually retired. I spent the early years in mapping the genome of cancer and then working on the RNA side, and decided I was going to turn it over to the young people of the world. And then I realized that this problem still exists. And so the challenge we have, not just in Italy, but around the world, is how do we reduce the cost of drugs? And then how do we have better diagnostics that can be portable, so that the right patient gets the right drug at the beginning of the disease and not at the end like they do today when it's impossible to help the patient. In most solid tumors, 70+% of the patient population actually will cure, I don't like that word very often, but at least become chronic if you catch it stage 1 or 2A. You catch it 2B, and it goes down to about 50%, and if you get to 3, it's less than 30%. So the challenge of early diagnosis is still there, but the importance of matching the diagnosis to the appropriate drug and having an affordable drug with minimal side effects is really a quest for all of us. And I asked myself this question: why is it, and now we've heard the story of SARS-CoV-2, we've heard the story of other diseases that the folks here are working on, but why is it 20 years after the Human Genome Project, we still cannot identify the appropriate way to manage a cancer patient? And I took a quote from one of my favorite books. Siddhartha Mukherjee wrote a book called The Emperor of All Maladies, about the history of cancer and how we've evolved to treat cancer. And this quote is very relevant. Down to their innate molecular core, cancer cells are hyperactive, survival endowed, scrappy, inventive copies of ourselves. In another place in the book, he says the only purpose a cancer cell has in life is to survive. And so because of their ability to think and see, we've now discovered some new capabilities that I think will really allow us in the next 10 years to dwarf the last 50 years of discovery in cancer. And really it's centered all around, thank you. I actually thought that was you trying to, so there you go. But really it's this convergence of understanding the tumor microenvironment, which has been a recent discovery that I'm very proud that Oncocyte has led and patented, with the evolution of spatial biology, being able to look at a 3D image of not only the tumor itself, like you might get from an image from a radiology or MRI, but also at the cellular level, to be able to look at the spatial interaction of components of the microenvironment, and then to use these large-scale artificial intelligent engines to synthesize and understand the right treatment first, and then ultimately be able to monitor in real time. So when we treat a patient and we know that we're going to see that tumor outsmart the immune system, how do we know when that moment in time is so that we can change the therapy, much like we did in my young years? I was very fortunate to run Roche PCR World in the early days of HIV. And the way we were able to take HIV from a deadly disease to a chronic disorder was learning how to monitor very rapid changes in viral of the viral code and re-drug the patient with a new cocktail to bring down viral load. The hypothesis that now is becoming proven in the US and I think around the world is that we can do the same thing with cancer. We can understand the tumor microenvironment and the interaction of the biology. With AI engines, we can predict what that biology will do in the next year, and then we can stay ahead of it by monitoring in blood what is happening over time, and then treat the new tumor before it becomes an actual metastatic event. And so I'll pause there because I think that I don't want to spend, I'm the only thing between you guys and a glass of wine, and I know that's an important end result. But I want to thank everyone. Diego, thank you for inviting me. And the notes I took today and the work that's going on in Trieste is quite powerful. And I think hopefully the outcome of COVID is we do better international collaboration. There's many things going on here that I didn't even know about till you shared, and now that I've been here and seen today. And I think that the evidence is that the need to democratize care and decentralize care, obviously in cancer, but also in the infectious disease world and some of the things Alessandro had talked about, is really powerful. And with the idea that we can look at a saliva sample which becomes transportable and a sample that will be okay a week from now, allows us to hope that we can solve some of these great challenges around the world sooner than later. And I do believe the next 10 years, with the right investment and the right collaboration, will really change the course of medicine, thanks to, unfortunately, a pandemic, but forced us all to work together to solve a global problem. And now we've figured out ways to do that together. So thank you again, Diego, for inviting me, and thank you for a few minutes to speak. Thank you.
Thank you very much, Ronnie. Do we have some questions or comments? Linia?
I was intrigued about the panels you found in circulating DNA and whether you matched this to genetic predisposition and whether, you know, it will have a fingerprint that kind of approximates the likelihood to have a certain disease.
Yeah, you know, my work is on the somatic side, but there are people working on the germline as well. And what I know in oncology is only 15% of cancers are actually hereditary or familial. The rest are environmentally caused by something, you know, could it be water, could it be hormones we take or drinking our milk, all those things. So our work is really looking in the blood and seeing and looking at epigenetic and small switches that we know turn on in RNA that are coding for these proteins. And so our work is really focused on a few cancers, obviously lung cancer, ovarian, which today is a horrible disease, pancreatic today, which is a horrible disease, where we can look in blood and we've synthesized the RNAs that are actually driving those tumors, and we can identify in blood in 24 hours if those signatures are active, which allows us to really rapidly identify an onset of an ovarian and pancreatic cancer. My challenge, Lavinia, and I think all of our challenges, it's an experiment that there's no retrospective biobank for. It's longitudinal, which means I can't petition my government for funding nor for regulatory approval until I prove it out, which means we have to unfortunately monitor over 3 to 5 years. And as you might imagine, in the US, academics are very proud of their information. So they don't like to share with us in industry. I see much greater collaboration in Europe between industry and the academic world, and which is why I'm in Europe now. I'm trying to generate some of that opportunity for us. We have a lab, as you know, in Germany, and we're trying to do some work in Europe, and eager to spend more time here because there's an openness here. The US, the academics, they're wonderful people, but they're very protective of their discoveries. So.
Yeah. Okay, thank you.
I'm Ronnie, really happy to hear that the industry is willing to share as well, because it's a both way.
It is both ways. I would tell you the challenge we've had in the past is large companies like Illumina and Thermo Fisher. I ran their global genetics business for a number of years, and one of the reasons I stepped away is they were not sharing the patents, they were not sharing the capabilities. And in my world at Roche, when I ran PCR, we licensed PCR to everyone, and the academics, we didn't charge very much for it. We charged industry a lot, but not the academics. And that led to this proliferation of a technology that's done a lot in the world. And why we don't do that for next-gen sequencing, it's one of the things I pound the table, and when I'm on Capitol Hill in front of Congress and things, we have to change this, but Illumina makes too much money.
Yeah.
So.
Okay. Thank you very much. So I would like to close this session on the research for biotech and energy high-tech innovation. I just prepared a couple of slides, like a conclusion before I pass word to Ronnie. So it was really a pleasure to be here with you and to chair this session. And I want just to summarize that today most of the talks were in the field of the research for fast, innovative diagnostics, accessible and affordable therapy, prevention and well-being. That's great news to hear that here regionally, nationally and internationally there is a lot of work going on. And we heard work on the diagnostics of different viruses, of antivirals, method for virus inactivation, prevention, detection, therapy. We heard about the hormone dimension and all the others, proprietary saliva testing models. Insulin biosimilars, strain and process development, design, development, production and characterization of different proteins in diagnostic tests. We talk a lot about the preventive personalized well-being, so that's a great thing. So we really start to think on the prevention and well-being. We heard about new ideas in the area of treatment of cancer like glioblastoma, and we heard all the challenges in oncology, monitoring tumor microenvironment, monitoring of therapeutic efficacy from Ronnie. Then we moved and we had the representative, and we heard this high-tech innovation to give a support to these healthcare systems through the advanced air mobility system, long-distance activities for transportation of organs, blood, emergency medicine, and we are happy that we have it here in the region, this really new advanced air mobility healthcare. And we heard also a little bit of the energy high-tech innovation that we hopefully more will be in the future, collaboration here in the region and nationally. We heard it from a representative of biogas services. So all of this, I just go back to my first slide. So we all somehow connected with this really with this industry 4.0, because it gains momentum through this intensification. So every single presentation today was about intensification, about predictive modeling, about data-driven decision that really opens opportunities to efficiency, strategic planning and agility within the industry. So really all of this was guided process in a more efficient and knowledge-based way. So we all agree through 11 presentation here, and we work on a better, smaller, easier, faster, and real time. If we talk about prevention, diagnostics, therapy, or whatever. So really I would like to wrap up to say that all of us that we heard that we work in this fast and reliable early diagnostics and therapy, and what is even more exciting, in a prevention and well-being field. So I would like to thank you all for your attention, and I will pass the word to Ronnie.
No, I just want to say thank you again. Thanks for putting up with my Italian, which is not very good. And thank you, Natasha, for letting me share the stage. And I just want to encourage BioValley and the folks investing in this area. You have lots of important initiatives that the world needs, and so I would just encourage you, don't give up. Keep focused and keep charging ahead. So thank you again for having me.